Hello, everybody. My name is Michael So, and I'm one of the Nautilus administrators here at the Inova Translational Medicine Institute. I see a few people are still making their way in out of the, gosh, it looks like we have about 20 some odd people that have registered. I'm going to give a few people, just give people about 5-10 minutes to get in. I'm not going to wait too long, though. I don't want to continue to encourage this behavior. Joining us on our list here, we have a number of people. I actually saw that Gerald McCain from ThermoFisher is joining us here today. So if there's something in here that isn't factually correct, I profusely apologize. I'm trying to present the material in the best way that I know it, and there might be better ways to do it. In fact, that's going to be something I'm going to repeat throughout the course of this presentation here, is that I don't consider myself an expert. If anything, I consider myself a hacker and just someone that has learned Nautilus on the way here. So if you guys have a better way to do this, or you have a topic that you would like to present, I would very much appreciate either the idea or someone to actually present. I am running out of ideas, so it's not going to be long before I say, okay, well, I don't know what else I don't know what else to present on, and we'll encourage you guys to put in your ideas just to keep things going here. I see more and more people are joining us here. I'm going to go ahead and continue to mute myself as people come in, and if you haven't already, please feel free to introduce yourself in the chat window. The chat window can be brought in. So I'm going to play this GIF here on a loop. We're going to be using the chat tool quite heavily. If everybody comes, we'll having about 20 some odd people joining us here today. So we'll be heavily using the chat. So if you look at the GIF here and how to remove or pull out that chat window onto your desktop so that you don't have to have the whole window taking up the real estate on your screen, you can do so. Also, for the benefit of people that won't be able to join us, this session is being recorded. And so feel free to, if you need to, change your name, but I don't think names should pop up here on the screen as it's being recorded. But we'll also be accepting questions either in the chat window or in this URL that's being shown at the top. And so I don't think you can actually click this, but what I can do is I'll copy this link and I'll put it in our chat window here. And feel free to ask any questions there. The benefit of doing the questions via this URL is that the questions will stay with the session. And at the end, I should be able to publish both the slides as well as any questions and answers that you guys might have. So yeah, we'll just give people 1 or 2 minutes here to get settled in. And again, we'll be using this chat window, so feel free to pull that out of your window, and we'll get started in here in a few minutes. All right, I'm going to go ahead and get started. So welcome to the ThermoFisher online meetup. We're trying to do this once every other month. So right now it's falling on the even months here. So our next one will be sometime in April, and the date will be chosen at some point, probably within the next week or two, and we'll send out an email for everyone that registered here today, along with on the forum post. So thank you for joining us. And just as a reminder, I don't work for ThermoFisher. I'm not a ThermoFisher employee. I know we do have someone here from ThermoFisher, but you know, I'm not here to endorse or they're not here to endorse or anything like that. I'm really putting this on to kind of share some knowledge that we've obtained from working with Nautilus. And as I shared before, for people that didn't come in, I'm looking for people that are willing to present on something that they've learned about Nautilus. It doesn't have to be perfect. You don't have to be a great presenter. If you're really nervous about it and you don't want to do it, but you have an idea of some topics that we can cover, feel free to, when we solicit feedback, provide that topic, because I'm running out of ideas. And I'm running out of things to talk about, and there's a number of things that I'm sure that other people are doing that we would benefit in learning from. I don't believe that I have the monopoly on good ideas. And I really hope that other people can take some of the things that I'm presenting on, either improve upon it or use it themselves. But ideally, you know, I really would like, you know, other people to be able to speak up and say, hey, that's what we're doing, or hey, have you thought about doing it a different way? Because that's what ultimately helps me and hopefully everyone else that's using Nautilus. And that's ultimately the goal. So just some quick notes here. We're going to be heavily utilizing the chat tool that's part of GoToMeeting. And obviously, we have a number of people here joining us. If you look at the attendee list, we have about 12. We have about a total of about 20 some odd people that are registered. So obviously, talking is not going to be something that we can easily do or easily accommodate over GoToMeeting. So we're going to be using that chat feature. Feel free to detach it. For whatever reason, this video doesn't want to load. But if you have trouble doing so, basically there's a little, I guess, dashed lines on the left side of the menu that you can pull out, and then you can have that chat window showing. So as part of a test, whether you're paying attention and making sure that my mic works and you guys can understand what it is that I'm saying, go ahead in that chat window, type a 1. Just let me know that you're there, and just so that I can see that everyone's there. So I see a number of people giving those ones. Thank you very much for doing that. That helps me to know that my message is getting across. You guys can hear me okay. So thank you very much for doing that. So we're going to talk about custom fields today. I get the sense that we're not the only ones that are using custom fields. The way that custom fields, at least that's been given to me and that explained to me, is that it allows you to capture information that Nautilus may not capture or may not have a field for out of the box. So you can make the fields work the way that you want it to work. That's important for your organization, the way that if you have a standard set of information that you want to make sure that every aliquot has or every plate has or anything like that, you can create as many custom fields as your database can handle, or if you have a separate DBA, your DBA will let you have. And it's great for capturing required information that's across entities, especially if this information is needed for 80, 90, 100% of the time. So like, for example, one of the required fields that we have in our system is what's called a collection time. And we didn't really feel that it fit into any of the default fields, because I know that there's like created time, there's also completed time, but none of those really fit in terms of our use case. So we created a custom field for collection time, and that collection time is required for any aliquot that's coming in that's a part of our research study. And so that's an example of where that creating that custom field can be useful. The difficulty, at least in my experience with working in Nautilus, is that it can be frustrating if you have a very small subset of aliquots that you want to track specific information, but for whatever reason, it doesn't fit in with the rest of your workflow. So for example, let's say you have about 100 aliquots that you want to attach like reagent information. or you want to attach, like, for example, what incubator it was in, because for whatever reason in your workflow, that information is crucial to understand and to have a part of your data set. So creating the column, at the end of the day, yes, it's relatively simple to do. You just go into the field creator and you create the field, it gets attached to the table. But if you do that for every single edge case that you have, you're quickly going to create a lot more columns than really you should ever need, and that can be difficult. So it increases your database table space usage, especially if you're doing this across a lot of different entities. And if you end up consolidating or you end up not needing a particular column, they can't easily be deprecated. Now, I know that ThermoFisher has a knowledge base article specifically designed for this. If you do not have that knowledge base or you're not sure where that is, get in touch with your support team, and they're more than happy to send that to you. But it's incredibly cumbersome to do. There's something about 15, 20 some odd steps, and it's very easily, you could easily mess something up along that process. It's obvious from at least ThermoFisher's standpoint that it was never intended to be deleted, and there are, I would imagine, very few cases where you could do that. Now, I would say in my 5 some odd years of being here, I've deleted probably 2 or 3 fields that were obviously created by mistake. These were fields that were never used at all. They had spelling mistakes, so it was obvious that they didn't intend it for to be there, but this never got deleted. And just from the cleanliness standpoint, just making sure that when you're doing queries and things like that, it can just take up space, a visual space anyway, where you're looking at all of the data, and for whatever reason, it just doesn't make any sense. And so it's also hard when you have that small subset of data. I know for me, my fallback is, well, is there a column that we can utilize so we don't have to create that column. And the problem with that is, and then you don't remember which column it is because it's not obviously named, and then you have to kind of remember if anything else is using it, and that you can fall into a lot of problems there. And I'm saying a lot of these things from personal experience. So let's just take a quick poll here in that chat window. There's some values here on the screen, so go ahead and type in your response. And let me know how many custom fields you guys are using. I imagine that no one here is going to be a 0. But if you've got 0 fields, go ahead and type a 0. If you've got at least 1, between 1 and 10, type a 1. If you've got more than that, you can see the values here on the screen. My guess is that a lot of people are going to be in this 2 to 4 range. I would say for us, we're probably in this 40 range. This is across all entities. So this is not just in aliquots or plate or anything like that, but we definitely have quite a good set of custom fields. John O'Neill here is saying that they have 50 to 300. I think we're pretty close to that. I didn't do a count. I was actually curious when I was writing this presentation. And I didn't do the count, but I imagine that we're pretty close to you, John. But what I find interesting in your response, John, is the range. You're not sure whether it's 50 or 300. I think that's great. I mean, obviously, everyone's using a custom field. I didn't see anyone write a 0. Everyone's using them, but my guess is that for people that are in the 20 to 40 or more range, is that this is going to get very easily something that you can lose track of. So the reason where this was born, where this discussion was born, was Rachel and I were. Rachel is in our chat room here. She's my a senior developer, and one of the challenges that we had with rolling out 16S extraction in Nautilus was, how do you capture things like the type of media that cell culture was in, or the reagent types and things like that. And if I'm being completely honest, I don't remember all the requirements, but there was a need for a number of custom fields, and what I did not want to do was add more, add any more than I needed to. And so, we came up with a couple of solutions that solve this problem. And there's 3 of them that I would like to share. We don't use all 3, but we use a combination of, I think, the method 1 and method 2 that seem to capture the majority of our cases. So this one is relatively obvious, where you create a field called metadata. And so in this case, we did metadata 1 name, metadata 1 value, metadata 2 name, metadata 2 value, and so on. And so you can create as many of these as you like. I would not recommend more than a few, because then you're just going to fall into the same trap that you did with custom fields. But the pros for this is if you have a small subset of data, let's say you have 100 aliquots out of the hundreds of thousands of aliquots that you have that need a special marker to indicate what kind of aliquot it is, or just additional information that you don't want to create a custom field for. You can say in metadata 1, you could say I don't think I didn't put an example here. You would say metadata 1 name is equal to reagent, and then metadata 1 value would be, for example, RNA later or EDTA. That way, you can track the values by saying in a where statement, you say metadata 1 name equal to reagent, and metadata value is equal to whatever. This schema is actually very similar. I actually took this from how WordPress does their tags. And so if you guys are familiar with WordPress, or if you ever developed the database for WordPress, this is how they do tags. So they say, the name of the tag, and then say, where that tag exists. And so, it's a very similar schema. And the beauty of this is that if you want to do different types across different entities, you don't have to create a custom field for each one. You can just use metadata 1 and say, this is what this is. And then drop that value into metadata value. It's very easy to customize. You can create as many as you need, but again, I wouldn't do more than a few, because otherwise you're just falling into the same trap. And the other problem with this is now you're doing 2 value names for each type. Now the benefit of this, though, is that if you're only capturing 1 or 2 or 3 metadata values, and those types and those names are changing between each aliquot, then this actually saves you space. This saves you effort in the database of creating more columns than you might need, because then you don't have extra rows or extra columns in places where that column is not necessary. I'm hoping that this schema makes sense, because I thought I had put an example here, but what I can do is, let me just throw up Notepad here. So if you were interested in, let's say, the name of the incubator, for example, so you would say metadata 1 is equal to incubator name, and then metadata 1 value would be the name of that incubator. And so let's say we have the incubator name oven 1. And then for every aliquot that you need this information, you would just iterate that through each one. And so let's say in metadata 2, you want to have the protocol that was used in that incubator. So let's say we would say human body temperature protocol. I'm sorry, this would be incubation protocol. And then there you would have human body temperature. And again, you would just iterate this for every aliquot that exists for that particular thing. And if you had another, let's say another set of aliquots that you didn't care about incubation name or incubation protocol, but you cared more about, let's say the person that it came from, you could do you could just change the metadata name and value. Now, Sean is mentioning a trick that you're actually spoiling the surprise, because that's method number 2, but I'll get to that in a bit. This we found to be useful in areas where you want users to be able to input data when they're accessioning the aliquot. So the beauty of this is that you can have these data fields be available to the user within the workflow. So this is method one. It doesn't require. I know that 9.3 makes some changes to how workflows actually work, so this might not apply in 9.3. But in 9.1 world, if you want the aliquot to be accessioned a certain way, you can have these metadata metadata one values to be available in your workflow and in your template. The cons of this is that you can't link the entities within Nautilus. So if, for example, you want to use for whatever reason, let's say you had another plate ID that you were interested in storing, you could do, a plate ID as metadata name and the actual plate ID in the value. Nautilus you can't have Nautilus link those two because you would have to create a entity link in the table. And since these values change, it wouldn't be smart to do so. Of course, though, you can always do joins as necessary within a database. So that's another way to do it. The other thing that we found to be it's not a huge challenge, is primarily we have a documentation area where we're starting to keep this information. You have to keep track of the metadata. So if you're using incubator name for one value and you accidentally put it in metadata 2, your data is going to get really dirty real fast. So having that consistent across the board is going to be incredibly important. And since there isn't any key in Nautilus to make sure that that's always the case, it's very possible to get this dirty real fast. And so I would suggest making sure that if you guys are doing any sort of code review, that you would make sure that those values are being incorporated. I'm going to read some questions here that I received through the Google thing here. John O'Neill is asking, have you considered removing the column from the entity so it's not displayed in Nautilus application interface? The entity properties allows you to order and remove columns so that they aren't available to users to see. So John, that's actually a very good question. So yes, I have considered simply just hiding the information in Nautilus. The problem comes down when you're doing any sort of data extract to another system, or if you're trying to link data with another system. So we do a lot of import-export with our internal data warehouse and with other customers elsewhere. And what's been problematic is, especially if you do a select star, is that there's column fields that are misspelled. Let's say they were made by accident, or columns that just don't really relate to the data. It can be really difficult to see, especially on a 17-inch or even 22-inch screen. So I personally have a 22-inch screen, and I probably abuse select star in the database more than I probably should. But if I'm not sure where a particular data sits, primarily because our data tends to not be consistent across the board, I found that that's a lot easier. And if there are columns that just aren't being used, maybe it's my OCE that says it need better to just remove the column. And so that's kind of where I sit. Now I know when I presented at ThermoFisher, I was in I got the looks of shock when people found out, or when I discussed how we do delete some level of data. A lot of people said, it's a lot better. You should be hiding that information. And I understand the argument, and to a certain extent, I agree with it. And it's something that that Rachel and I have actually discussed, in whether we should change the methodology to accommodate that. And so for right now, I would say that our SOP right now is still to delete rows and to delete columns in this case. But we are trying to find better ways to do it. And obviously, your question is very good at that. And so let me go back to our slides here. So let's go to method 2. And Sean made the great observation. It sounds like someone else has done this, and we're not the only ones that discovered this. Is I would like to call this tag-based. So this is a little bit more consistent across Nautilus because you can have it be in the workflow, and it can be a drop-down that users can add. The problem that I've had with this particular method is that it can only work for areas where where a test workflow can be attached. As far as I know, it's only aliquots and tests that, aliquot templates that can have a test workflow. I think that SDGs or something like that, so one that we don't use, has the ability to add tests, but if you want more information in an area where it isn't, where it isn't a plate and it isn't an aliquot, then you're kind of stuck with method 1. But we actually started using this test result-based the storing of information when it wasn't when it was directly related to the aliquot in question. So the example that I'm pointing out here is that we have a tissue container that is collected outside of Nautilus and then brought in, and so it's not happening live. It usually happens about 40 to 60 minutes outside of Nautilus, and we want to be able to capture that information. And so we want to be able to say what liquid nitrogen dewar was it in, what time was it stored. And so obviously this isn't universal across anything that we're doing. And so what we say is that, okay, once you accession the aliquots, attach this test and just give us the information of the dewar and the time that it was stored. And the pros for this is that since it's Nautilus based, it really does allow you to use the workflows the way that they are intended. But the test result, what I have noticed is that it's kind of a bit of a hack, but I feel a little bit better that other people are using it, where you can kind of capture details about an aliquot that isn't strictly a test. So for those that may not be using tests, it would be things like a QC value, what it's traditionally for. So if you have a QC value, like for us, we have a nanodrop QC for our DNA extractions. You're interested in that 280 over whatever value it is, and you want to capture that number. And especially when you're talking with a laboratory personnel, tests makes sense for them in that case, that QC value. So if I'm being honest, we did method 2 first. This came about probably about 2-3 years ago when we started doing this very heavily. And it wasn't until we did the cell culture where we really wanted to capture information upon login that we built the metadata columns. Because in order to do the test, the aliquot needs to be accessioned first, and then the test attached, which isn't ideal in some cases. And so I think it depends on the action that you're trying to create and what the data that you're trying to capture. So just out of curiosity, Sean, I'm going to unmute you here. Do you use this in a similar fashion in Nautilus?
Yeah, we normally tend to use it to for information associated with our tests. One thing that they want to start doing is they want to capture what time they're extracting everything, and it doesn't always fit into other fields. So it's easier to make a new result template for it and then put it in there. And that way it's still linked to everything, and I'm not having to add new columns.
Oh, that's perfect. So just out of curiosity, when did you guys start doing it that way?
That was somebody that was started by my predecessor, so I do not know exactly. But I've been working with Nautilus for about 6 years now.
Okay, very cool. So I'm glad that someone smarter than me figured that out. Thanks, Sean. So yeah, I think I think both methods, both method 1 and 2, have their pros and cons as to why you would do one way or another. And so again, these slides will be available after this presentation, and hopefully, if you're not doing this now, hopefully this will help you. If anything, it'll make your database, your DBA, very happy. One of the other things that I forgot to add here in terms of the pros, from a database standpoint, the way that this works, and the reason why I call this tag-based, is that the way that the Nautilus stores the test name, the result name, and the result value is very easily scalable. So they're all individual rows within the database. They're not additional columns, which is great. So in many ways, what method 1 does, it mimics method 2, except the difference being that you can bring data upon login. Under method 2, the rows or the particular value that you're interested in is created as just additional rows within the database. So there's no column creation there, and so again, it's incredibly scalable. As long as you understand the various caveats of doing it as a test. The other con here that we found, at least with our users, is that when you do a test and you haven't modified the aliquot color behavior, it'll turn red and yellow, and people, at least in our lab, really get freaked out by that. And it's something that we have discussed changing to better reflect what these colors mean. Right now, what we tell people is that the colors don't mean anything, and you should ignore them for now. We eventually do want to make them mean something, like the red actually means, hey, there's something wrong. But I think right now, my understanding is that the way that Nautilus does the colors is that red means that it's complete and you should stop. There's no more work to be done. It means stop. Yellow means something is in progress, and green means, hey, there's stuff here for you to do. And so that's my understanding of what those colors mean. All right, method 3. I don't use this, but we have discussed using it. This is XML JSON-based storage. I would think that this is probably going to be more helpful in 9.3 than in 9.1. 9.3 allows you to have clobs, which are unrestricted column names. And so I believe the limit in Nautilus is 2,000. The limit in the database is 4,000. How the two reconcile is a little bit beyond me. But in any case, you could use XML or JSON to store this information in a way that is not available. This is actually a trick that I used to use back when I was back when I did web development work and I didn't have the ability to create columns. I just made a column, a text field, or that was a text field, and just used it to store additional information. The problem with this is that this is very, very cumbersome. It can only be parsed by the database and is not natively supported in Nautilus. So if you want to report back data in an XML or in a JSON format, you've got to parse it. And there's no easy way to do that in Nautilus. The other part is that it can be slow and cumbersome if you don't have the schema prepared correctly. So the example that I'm providing here is kind of a use case where we wanted to be able to scan or at least encode some aliquot information into a barcode. So the goal here was to make a QR barcode with this information in it, have Nautilus and parse, or not Nautilus, the database parse the information and fill in the information where it could. So it would say that the barcode was 123 ABC, and then it would add these additives and reagents as either custom fields or as additional hazards, I think it is, in the aliquot. And so that's one of the beauties that that the XML JSON provides, is that you can encode it into a barcode and have a lot of data in a very small amount of space, and then have that data stored within the database. Like I said, I don't use this currently in Nautilus. I can't. It is, I believe, is a little bit over-engineered, but I could think of some use cases where this might be useful. I mean, outside of the example that I described. Before I continue on, are there any questions that I can try to answer? I'm hoping that this makes sense. So if you don't mind tagging into that chat box, giving me a exclamation point if it makes sense, question mark if it does not. Very good. Looks like everyone is getting this. All right. Well, that's pretty much it. I didn't want to go too far in depth with those methods. The way that I would describe a lot of that, a lot of the custom fields, they're really hacks. We're doing things that Nautilus may not have been designed to do, but it helps you to capture additional information. If you want to present or you have an idea for a future topic, please fill out our feedback and our feedback form and let me know. You can reach me at michael.so@inova.org, and you can look at my LinkedIn profile right there. You can take a picture of that barcode with your phone, and it'll take you to a web page where you can fill out some feedback and let me know how we did. My goal is to provide as best and accurate of information as I can. Again, I'm not a Nautilus expert. I've just been working with it for about 5 years. Sounds like Sean has been doing it a little bit longer than I have, which I certainly appreciate his comments here today. And so yeah, if you guys don't have any questions, that's pretty much the end of it. That's all I've got today. Again, if you have more ideas, please let me know. I will stick around here to answer any questions that people might have. I can unlock or open up our lines here. So if you guys want to chat, you guys can do so. But thank you guys for coming and participating in our second meetup. We'll have another one in April, probably about the same time, second or third week of April.